Welcome to everybody who's here in Missouri and to all of those who are listening by the video conference. Yes, each year we do ask the scientific directors and other experts to nominate individuals that they feel have represented this model of the astute clinician, physicians who have, through their own experience, identified particularly interesting and important challenging problems and then have worked that out through a wide variety of methodologies and approaches. And today's presenter is a wonderful example of that same approach that over the years has taught us so much about so many things in medicine and will no doubt continue to do so. It's my particular pleasure to introduce this speaker because he and I happen to know each other pretty well. In fact, I think we met on the first or the second or maybe the third day of medical school in the same gross anatomy room, dissecting cadavers and trying to maintain ourselves with some degree of equanimity, and a lack of vomiting would have been good too. And yet over the course of those months, I grew to find out during those 4 years of medical school that Ron Falk was an exceptionally intelligent, thoughtful, hardworking, dedicated guy who was going to be undoubtedly a leading physician in our nation. And boy, did that turn out to be true, as you will hear in a second. Not only did Ron and I do medical school together, we both then stayed on as the house staff at the University of North Carolina in North Carolina Memorial Hospital and worked side by side as interns and residents in internal medicine. And happy to say also that in that residency period, had the chance to meet someone who became his future wife, who's also sitting here in the front row, Dr. Catherine Falk, who also is a dear friend of myself and my wife, Diane. And we're particularly honored today that not only the two of them are here, but their daughter, Christine, who is now a second-year internal medicine resident, has joined us. The connection doesn't end there, because my daughter, who grew up in Chapel Hill, fell in love with medicine, decided she also wanted to be trained as a physician. Where do you suppose she went into medical school? Well, the University of North Carolina. And what particular specialty did she choose? Internal medicine. And then what subspecialty did she choose? Nephrology. Well, who was her division chief? Dr. Ron Falk. So over the generations, the Collinses and the Falks have had these connections, and it's wonderful to have yet another one here this afternoon at NIH. We're really pleased that Ron is able to be here to talk about this particularly interesting topic that has been his life's work, and which he has essentially created an entire field of endeavor around this thing called ANCA vasculitis. I got to say, Ron, when we were medical students studying the various aspects of human medicine, the area that I found of all the areas of disease, and particularly pathology, that it was most confusing, it was glomerulonephritis. And yet somehow you decided that that was the place where you wanted to land and where you have made remarkable contributions. I guess that just says we're a little different. I decided to do something simple like reading DNA, and it seemed a lot more straightforward. Well, in addition to all the work that he has done in the research area, he's also a fellow of the American College of Physicians. He has been a member and a chair of numerous NIH study sections. Thank you, Ron, for your service. And after rising through the ranks at UNC, first assistant professor, then associate professor, then professor and chair of nephrology division and hypertension, he has now, for the last year, become the chair of the Department of Internal Medicine itself. So without further ado, and without slowing down the process anymore, but I just had to say a few of those things, please join me in welcoming Dr. Ronald J. Falk.
It is indeed a pleasure and honor to be here. I must digress for a moment, though. Francis was my chief resident when I was a resident. And in medical school, the hardest subject for me was aminoacidurias. And believe it or not, as a medical student, Francis had come up with a slick way of identifying all the aminoacidurias. And so when one had research presentations, all of us would have pretty mundane sorts of things, and there was Francis with an approach to quickly identifying every aminoaciduria that there was, so there was never a question of who was going to go where. So being this astute clinician is both a tremendous honor. It is a wonderful and interesting opportunity, and it's a humbling experience. It's a humbling experience because what I'm about to try to do is to explain to you what has driven our research. And what has driven a lot of our research is, in fact, the questions that emanate from the patients that I typically see virtually all Thursdays of my life. We have a clinic that takes care of patients with glomerular disease and vasculitis. And I would tell you over the course of these 30 years and literally thousands of patients, the common questions that I get are, what's the name of my disease? How did I get it? What's the real driving cause? And then an associated question that is, what will make it go away? What will make it worse? What will make it better? What's involved in disease pathogenesis? And then finally, once we have managed to get somebody under control, in remission, the next usual questions are, okay, what would promote a relapse? And even more importantly, what sustains a remission? And is a cure or restoration of health possible? Restoration of health, a different word choice, and that one can imagine that one has cured a patient, but the long-term sequelae of what one's done to them may not have restored their health. So these are the questions that have driven our research, and where we began was with a case, a 15-year-old, taken care of by Richard Morris, Francis, if you would recall that name, who had the sudden onset of coughing up blood and a previously healthy child with a 2-week history of a flu-like illness, and on exam, it was hypertensive, had pulmonary findings of diffuse crackles and some pulmonary bleeding. Her creatinine was 10, and at that point in time, we had no clue what she had. She was treated with the usual course of methylprednisolone, and we took this young woman's blood, and because of an article that I'll show you in a minute from Lancet, we took her blood and dribbled them on my colleague Charles Jeanette's neutrophils, and lo and behold, saw this interesting staining pattern, which we now know as anti-neutrophil cytoplasmic autoantibodies. And this particular individual had a cytoplasmic or proteinase 3 derived autoantibody, and quite quickly we realized that there was another serotype known as perinuclear, or the antigen myeloperoxidase ANCA. Fast forward to 2016, these serological tests are ordered almost too frequently. In fact, they are ordered too frequently. But they do allow the practicing clinician to perceive that an autoimmune vasculitis may be present. It should be noted that this early part of the ANCA story was first observed in England by a group of investigators studying Wegener's granulomatosis. Our own work promptly discovered that, in fact, the perinuclear ANCA was associated with myeloperoxidase in patients just like the young girl I described, who had a necrotizing and crescentic glomerulonephritis. And then the hunt was on to figure out the antigen specificity of cytoplasmic ANCA. Raoul Goldschmeding in the Netherlands figured out that it was a serine proteinase, and quite quickly thereafter, we and others described proteinase 3 ANCA. But in reality, the first observation was really made by a pathologist in Australia, who again was studying individual patients who had a form of necrotizing glomerulonephritis, and all of whom had the Ross River virus, arbovirus infection, and he probably really defined ANCA for the first time. The widespread testing and use of ANCA among practicing clinicians resulted in a cascade of important discoveries. First, a new classification system was described, and I'll show you that in a minute. And one could then begin to name these patients as either those who had ANCA vasculitis or ANCA-associated vasculitis. There was quickly the recognition that disparate clinical symptoms were really part and parcel of the same disease process. That phenomenon led to a whole slew of organized, randomized trials that simplified induction therapy and helped us understand maintenance therapy. A far cry from the history of vasculitis, in which, in fact, individuals were seeing and feeling vasculitic lesions. Pearl Zeek thinking that in her classification system, that in fact most patients had a hypersensitivity angiitis as a consequence of sulfa drugs. And then good old Jack Churg, a pathologist, again who was studying individual patients, realized that in fact these diseases, at that time called Wegener's granulomatosis, his disease, allergic angiitis and granulomatosis, and what was then called microscopic periarteritis nodosa were all related and all small blood vessel diseases. There have been 2 Chapel Hill nomenclature conferences. These have been really interesting and wonderful opportunities to get the international community together. And this nomenclature has now held sway. The ANCA-associated diseases that you see on the bottom, microscopic polyangiitis, we got rid of the term Wegener's granulomatosis since Friedrich Wegener was not a nice man, and now use the term granulomatosis with polyangiitis, and then instead of calling it the Churg-Strauss syndrome, calling it eosinophilic granulomatosis with polyangiitis. All of these are small vessel vasculitides and are distinct from the large vessel, middle-sized vessel, and even the immune complex diseases. When one looks at the pathology that's associated with these diseases, you can imagine that any blood vessel would be involved, from pulmonary capillaries to glomerulonephritis to venulitis in multiple organs, and in fact, many times small and even middle-sized arteritis. And the clinical phenotype then follows the course of where vessels are injured. What was also pretty quickly established was that these ANCA-associated diseases were different than those associated with immune complexes, those associated with linear staining along the basement membrane, otherwise known as antiglomerular basement membrane disease, but in fact were marked by the absence or the paucity of immune deposits, an observation that perplexed us, for in fact that observation was the beginning of the question of what actually was driving the pathogenesis of these disorders. So you can imagine then from a clinical perspective, if we line up the 2,000 or so patients that we have had the privilege of seeing, some individuals have organ-limited disease. The most common are the leukocytoclastic angioidities of the skin. Others have had peripheral neuropathies associated with mononeuritis multiplex, or unfortunately, transmural infarcts as a consequence of vasculitis of larger vessels in the abdomen. I'll talk to you a little bit more about my favorite organ, the kidney, in a minute. But the pulmonary disease, in fact, is probably the most interesting, for their patients have historically been divided into those individuals who've had pulmonary hemorrhage or hemoptysis versus those who have a more granulomatosis phenotype with nodules or cavities. But the grim reality is that the overlap between these two potentially different phenotypes is broad, and many patients have any number of these pulmonary manifestations concurrently or synchronously. The upper respiratory tract disease presents yet another interesting pathophysiologic and clinically diagnostic conundrum. The upper respiratory tract, of course, is involved. Sinusitis, otitis media, cranial nerve entrapment, subglottic stenosis. But it is, in fact, the nasal crusting and sinusitis that is by far and away the most common. And the question there is it an infection that is causing the sinusitis, or is it a vasculitis that is creating a substrate for infection to occur? And there's data to suggest that superantigen-containing Staph aureus in the nose may actually drive relapses. Many of our patients, if not most, have had a migratory polyarthropathy. And what has been interesting yet again is that 10% of our patients clot, and they clot as a consequence of yet another series of autoantibodies. If you spread out all of our patients, if you spread out patients with various organ manifestations, and here I'll show it to you in the kidney, you can imagine that there are patients who have explosive disease who have rapidly progressive glomerulonephritis, and others who have a little bit of blood or a little bit of protein with necrosis that comes and scars and comes again as an aggressive lesion and scars. This on-off relapsing and remitting disease, when not stopped in any organ system, leads to scar and end-stage disease. So there is, if you aggregate all of our patients, a tremendous spectrum of lesion. What is also curious about this disease is the focality of it. Look at this glomerulus. It's a beautiful structure. Here is a portion of a glomerulus that's been injured with a necrotizing lesion, and here is a portion of a glomerulus that is absolutely, at least from our current being able to see it, unscathed. Probably best even seen in the skin. Areas of the skin that are not harmed, and yet ulcers and palpable purpura. Why is this lesion, why are these lesions so focal? I'd love help in understanding that. But when one comes back, in fact, to these two serotypes, it turns out that one can actually classify patients not on the basis of their histology, not on the basis of words, microscopic polyangiitis or granulomatosis with polyangiitis, but one actually now can identify patients on the basis of whether they have proteinase 3 ANCA disease or myeloperoxidase ANCA disease, an absolutely simple and very useful division. And why? It turns out that there are real genetic differences between myeloperoxidase ANCA patients and proteinase 3 ANCA patients. These differences are largely and expectedly associated with HLA. The phenotype of patients with proteinase 3 ANCA and myeloperoxidase ANCA, while they overlap, is distinct. Patients with PR3 ANCA have much more upper respiratory and lower tract disease, whereas myeloperoxidase ANCA patients have much more kidney, skin, neural disease. And if you live in China, regardless of the disease descriptor, the majority of patients have myeloperoxidase ANCA disease largely because of that HLA. And from a clinical perspective, the reality is that patients with proteinase 3 ANCA relapse far more commonly than patients with myeloperoxidase ANCA, an important clinical observation, for in fact long-term therapy is less needed in those who relapse very infrequently. So in fact, in 2016, our thinking is that patients have myeloperoxidase ANCA disease or proteinase 3 ANCA disease, over the course of time, smaller and smaller group of patients who look the same but remain ANCA negative. And then one could have a series of descriptors, but the basic differences are on the basis of these two serological markers. The therapy of this disease was revolutionized by one of your own. Here you see a picture of Dr. Fauci getting his Presidential Medal of Freedom because of a disease that he migrated into rather than staying with his roots in autoimmunity and vasculitis. For his use of cyclophosphamide, of course, changed the lexicon of how we think of these diseases. And fast forward 50 years, the therapeutic jungle of possible therapies and the vines on the trees are large, from steroids to cyclophosphamide to methotrexate, azathioprine, mycophenolate mofetil, when to use plasmapheresis, and the new kid on the block, rituximab. The therapeutic options are substantial, and there have been a raft of organized trials that have helped us understand that oral cyclophosphamide that Dr. Fauci suggested in terms of maintenance, that azathioprine would work, that cyclophosphamide may be better than methotrexate for upper respiratory tract disease, that azathioprine was better than methotrexate, and azathioprine was better than mycophenolate mofetil. We've learned a lot as a consequence of being able to aggregate patients with the ANCA as the entry criteria. The induction therapy has also changed. For in patients with severe disease, the combination of rituximab and cyclophosphamide has proven to be a very useful approach, whereas in others, the removal of the B cells that contain CD19 and CD20 has proven to be a very useful approach in these antibody-induced disease. And finally, one can maintain patients in remission, especially if they're PR3 ANCA patients, with repetitive doses of Rituxan, a far cry from Anthony Fauci's days. And so where we are now is if the person comes in with pulmonary hemorrhage, that young lady, we would use plasmapheresis or methylprednisolone, which we're using less and less of because it causes diabetes, cyclophosphamide and or rituximab, then azathioprine or rituximab, and the real question is when to stop. And this has been remarkably effective, for in fact, our own long-term remission rates are pressing 93%. The real question is the relapse possibility from 0 to 50%. Who is 0 and who is 50%? And how can one predict which patient is going to do what? I continue to worry that rituximab takes a while to work. You can imagine a patient coming in, getting their rituximab, and the disease progressing, renal function declining, need for more medicine. And the real worry now is because we have so many tools that patients are presenting now with sepsis, with infection, and we are no longer at a stage where we're trying to put vasculitis in remission. We're at the stage of worrying that we're using far too many drugs. So in my mind, the first take-home message is that ANCA, which started as a biomarker of disease, is now the name of the disease. And it's resulting in earlier diagnosis, more prompt therapy, and much better long-term results. Okay then, what's going to make my disease go away? What makes it worse? What makes it better? And from the very beginning, when we saw that autoimmune glomerulonephritis, we asked ourselves the question, well, how do these neutrophils and monocytes get to the vessel wall? What's driving them there? It's not an immune complex. It's not anti-GBM. And so we began this hypothesis that in fact something must be making leukocytes marginate and adhere and diapedese, and then once there, degranulate and have a oxygen radical production, and that would then create necrosis and karyarexis, a model then that we have tried to prove for some time. There was clinical evidence that ANCA were pathogenic, the association, the correlation of ANCA titer with disease in some patients, the epitope specificity, and then there's a single case of mom to child placental transfer of the disease, and many examples of drug-induced vasculitis. But in 1990, we asked the question, if we took ANCA, either myeloperoxidase or proteinase 3, and incubated them with normal leukocytes, could we in fact see that there was a respiratory burst and degranulation? And in fact, one could. these autoantibodies interact with neutrophils and monocytes. And now, literally with hundreds of experiments and manuscripts from the international community, we all realize at this point that ANCA interact with the cell, neutrophils or monocytes, and that somehow or another, the antigens that are in granules now become expressed on the surface of the cell, and that then allow for antibody-antigen interaction with Fc receptor amplification. And at the end of the day, one can show that these activated cells on transwell plates cause leukocytes to adhere very specifically, marginate, and penetrate. But what caused the antigen to become exposed? And in our early studies, we used a number of cytokines that we showed in that original paper caused myeloperoxidase and proteinase 3 to come to the surface of the cell quite quickly. We have another thought of how these antigens get to the surface, and that has to do with epigenetic deregulation of the gene, and I'll talk to you about that in a moment. But the real breakthrough came, in fact, with animal models. There are 3 of them, and the question is, in each one of these models, how was tolerance broken? In the first of these, our own series of studies, we gave native mouse MPO to myeloperoxidase deficient mice. Another group of investigators gave human myeloperoxidase to rodents, to rats, and they too got a necrotizing vasculitis. And the third model, which in fact may be the best model not of pathogenesis but immunopathogenesis, was that a group in Australia, Kitchen and Holsworth, gave mouse myeloperoxidase to wild-type mice and caused the disease by adding a little bit of anti-glomerular basement membrane disease, or LPS. The 2 models that I'll talk to you about are the first, our own, and I'll come back to this Australian example in a moment. This is a simple set of experiments. You use mouse MPO, you give it to a myeloperoxidase knockout mouse, and the disease happens quite quickly. A beautiful example of vasculitis that in fact looks remarkably similar to the human disease, with fibrinoid necrosis and crescents and vasculitis in a number of organ systems. And we've learned a lot from this model. We've learned that the antibody all by itself can be passed from animal to animal and can cause the disease. We have fulfilled Koch's postulates. We know that neutrophils and monocytes are required. If you titillate a neutrophil, if you make it primed, if you give the animal a flu-like illness, that will exacerbate disease. The T cell by itself does not cause the disease. We'll come back to T cells. But what was really interesting was that the alternative pathway of complement really seems like it was hugely important in this disease, and in fact, in particular, the C5a receptor. And one could stop the disease in animals that did not have the C5a receptor. So in fact, we and others put the human C5a receptor and knocked that into mice and knocked out the mouse C5a receptor, and then using an oral antagonist, we were able to block disease. And quite frankly, now we are in not just a phase 2, but almost a phase 3 trial of this compound called CCX168 that is the exact same molecule in human ANCA vasculitis. And preliminarily, it looks like this drug may actually decrease hematuria. Take-home message number 2. ANCA really look like they are pathogenic, and they target circulating cells rather than targeting directly the vessel wall. It's an indirect form of autoimmunity. So how did I get this disease? You've told me its name, you've told me what makes it worse, you told me what make it better. You have a treatment approach, but how did I get it? Paul Ehrlich, many moons ago, described this phenomenon. And to quote him, it would be dysteleologic in the highest degree if self-poisons of the parenchyma, autotoxins, were formed, in which case we must be justified in speaking of a horror autotoxicus of the organism. Autoimmunity is uncommon because it's not, in most patients, a single hit. And one can imagine then that it's the accumulation of a number of hits that must be required in humans. There must be a genetic predisposition, and as I'll talk about in a moment, that's largely HLA. There must be defective T and B cell regulation, and I'll show you that data. There are, for sure, environmental factors that lead to the accumulation of neutrophils and monocytes that cause the disease. But what is it that allows some patients to resolve and others to progress and scar? What is it that allows some patients to relapse and others to have long-term remission? Well, I can tell you quite quickly, T-cell alterations in ANCA vasculitis are substantial. Yes, there are Th17 cells in the periphery. We and others have shown that they are specific for autoantigens. There's an increase in effector memory cells. And unfortunately, while there are more T regulatory cells, they are unable to suppress. And I'll show you why that's true. Most of you may not like to look at flow plots, but the abnormality in ANCA vasculitis is not subtle. Here is a effector CD25 low cell, T cell, in a healthy person. And most of you, I predict, would be sitting right here. Here are CD25 intermediate effector cells that proliferate wildly. They're there during disease activity, and they're there in disease remission, and they don't respond even to normal T regulatory cells in the same way that these peripheral T cells do. And to a certain extent, that's true because the Tregs, the T regulation part of this, is also perturbed in that these Tregs do not suppress. And interestingly, in ANCA patients, they don't suppress because most of their Tregs are exon 2 deficient. And that, we think, is why their regulatory capacity is altered. And there are a cascade of studies that would suggest that B regulatory cells are also altered with tremendously decreased IL-10 production. So take-home message number 3. There is a broad-based dysregulation of T and B cells, and that, of course, then provides the substrate for an autoimmune response in these patients. But coming back to the patient again, we've had multiple examples of patients whose ANCA titer fits beautifully with the disease course. And of course, I would have loved every patient to behave exactly like the big red line on the bottom. Not true. For there are many patients in whom commercial tests for ANCA would suggest the ANCA titer doesn't change at all, that it's still there, regardless of the clinical activity of the patient, and that prompted us to ask the question, well, why? And there are 2 explanations for why. First, we are looking on commercial tests with an antibody to the whole molecule. If, in fact, you look at specific epitopes using an epitope excision approach and mass spectrometry of individual patients, what you see is that in patients with active disease, they have a series of reactivity to specific epitopes, which go away when the patient's in remission. There are epitopes that are found in patients in remission, which these active patients, when they go into remission, still have. And then, as one can imagine, there are naturally occurring autoantibodies in many people to myeloperoxidase and proteinase 3 that are similarly there. And in fact, one of these epitopes we have found in patients with ANCA-negative disease. This B-cell epitope, called, which I'm showing you here, we've affectionately called it KIVI, is this B-cell epitope found in patients. At the same time we were doing these studies, our colleagues in Australia were asking the question in their mouse model, what actually was the important T-cell epitope? And it turns out the T-cell epitope is very much overlapping with our B-cell epitope. And what's equally interesting is that our B-cell epitope is hidden within the MPO molecule. It's not available to solvent, whereas the T-cell epitope is in fact exposed and is available for interaction, that small piece that doesn't overlap with the B-cell epitope. The Australians have taken this one step further, for now they've taken their T-cell epitope and have induced mucosal tolerance to the entire myeloperoxidase molecule. Can we do the same? And where we're headed here is we have sequenced HLA, we have in silico and then done binding studies, and have found a small group of patients who in fact have the human version of this same T-cell epitope and are getting ourselves in position with nanoparticles that permit just one kind of T-cell response to ask the question, can we do mucosal tolerance in humans? So the genetic basis of this disease, as you would imagine, is largely HLA associated, and that's what has turned out to be true. And we've used this HLA phenomenon for the studies I've just described. And yes, there are environmental pressures, seasonal variation, a cascade of drugs. Be careful when you're using cocaine that you don't cut it with levamisole. Pick your cocaine supplier carefully. But we use hydralazine and propylthiouracil. We're good at creating the disease ourselves. And silica exposure seems to be a pressure as well. But it's been remarkably difficult to prove that molecular mimicry is responsible for ANCA production. Where did I get my disease? How did it start? And we've been looking for molecules that could be a trigger and found, in fact, the opposite, that patients are reacting not to the sense peptide, but either on Ross River virus or Staph aureus or a number of microbes, that they're reacting to a complementary peptide that then generates an anti-idiotypic response that produces an ANCA. And we have shown this with autoantibody studies, with T-cell studies, with very specific HLA studies, and the new North American GWAS is going to show again that complementary PR3 is an important target. But what is driving my disease in those individuals whose ANCA titer remains and are in remission? That makes little sense. That issue perplexed us and still does. What is it about myeloperoxidase or proteinase 3? Is the molecule different, or is there something else going on with these molecules that's making the antigen available? An experiment by chance of serendipity, where Dr. Yang in our group showed that in fact proteinase 3 and myeloperoxidase message was present in mature neutrophils. Dr. Kaplan, if you're in the audience, yes, it could be low-density granulocytes. But in fact, there was no question that patients, not healthy controls, had turned on the genes for myeloperoxidase and proteinase 3 completely unexpectedly. And it's not subtle. We've looked at almost 1,000 patients. There is a real turning on of the proteinase 3 and the myeloperoxidase gene, and we've looked at a cascade of disease controls. This is not subtle. And what's more, in most, but not all patients, the proteinase 3 and myeloperoxidase message levels correlate pretty nicely with disease activity. Interestingly and unexpectedly, since myeloperoxidase and proteinase 3 are on 2 different chromosomes, there's a reasonable correlation between when the PR3 and when the myeloperoxidase gene are turned on. And we know from using clicket chemistry on patients' own neutrophils and monocytes that when the genes are turned on, there's new protein production. Not existing, new protein production. You're going to ask, or could ask, well, is that new antigen production? And we don't know the answer to that yet. But there's broad-based epigenetic dysregulation. It turns out that there are two forms of PR3, PR3-3 and PR3-2, and in some patients, 25% or so, this alternative transcript is turned on when patients have active disease and turned back off. This is not a subtle epigenetic dysregulation. And there are two potential reasons why this may be happening. One is a histone methylation event. Dominique Chivat and our group discovered that, in fact, the PRC2 complex, as a consequence of RUNX3, is brought both to the PR3 and MPO gene in very specific places. And in the absence of RUNX3, which occurs in these patients, the PRC2 complex is not brought to lysine H327, and the gene is turned on. But if this is really an important gene, it shouldn't just be histone methylation. There should, in fact, be DNA methylation. Remember, if you have an unmethylated gene, the gene is turned on, and if you have a methylated gene, the gene is usually turned off. What about patients? And in a study that's coming out on Friday, we looked across the PR3 and MPO genes for CPG islands and reefs and shoals and you name it, what kind of CPG we could look for. And here's the PR3 promoter, the transcription site. And in 1996, the PR3 transcription was shown to be tremendously influenced by this factor called PU1 and a very specific CG upstream of the PU1 site. And in fact, we've looked very specifically at that CG site. And if, in fact, a patient has methylated that CG, nothing else, their chances of relapsing, their chances of staying in remission over the course of almost 3 years, it's substantially better than if over the course of time they are unable to methylate that particular CG island, or in fact the methylation there goes down. In fact, I would argue then that it's not just the presence of the ANCA, but also epigenetic dysregulation of ANCA antigen genes that may be responsible for causing the disease. So finally then, what will prevent it from coming back? I wish I knew. We have now many, many patients, hundreds, who are in long-term remission off therapy, perish the thought, but they are for years. Who are they? Well, they don't have PR3 ANCA, most of them. They don't have respiratory tract disease, many of them. What is it that allows some patients to resolve and some patients to scar? That's a huge focus of our research. For I would dearly love a biomarker of remission. I would love a biomarker of relapse. The clinical biomarkers of relapse right now are proteinase 3 ANCA, upper and lower respiratory tract disease, or the converse. What's a biomarker of remission, and how important clinically is that? For if, in fact, you knew the patient was going to be in long-term remission, my worry of over-immunosuppression as a consequence of continued use of immunomodulatory drugs would be tremendously decreased. But it turns out, and now shown by the Vasculitis Research Consortium, that despite all of our efforts, the patient really, really does know best. They looked at a patient global assessment tool and captured how patients judge their own disease activity, and patients always can tell you 3 months ahead of time whether they're going to flare, whether they're flaring, or whether they're in remission. They know. And that's the next huge question. What do they know that we can't find a biomarker to tell them? Of course, if you really listen to the patient, they tell you, I don't need a biomarker, I know my own body. And that's an incredibly important lesson. Another approach that we've had to figuring out when patients relapse is a low-tech approach. We send everybody home with a dipstick. Peeing on a dipstick is fun once a week or once a month. You all can do it. If in fact, if a person has no blood in their urine and now has blood, we look at their urine quite quickly. We tell patients if their disease is flaring in whatever body part was initially involved to call us and we'll see them. Patient-physician interaction and patient engagement is really the ultimate response to this question. So if I were a scientist continuing to study this, and I still am, I think the whole question of relapse in remission is illustrated on this slide. What's driving the antibody to be produced, and what's causing the antigen to be turned on or off? Yes, I can make the antigen come on with a flu-like illness. Please make sure all your patients have had their influenza vaccines. But the epigenetic dysregulation is really a driving question. So our first case, this 15-year-old would have a PR3 ANCA vasculitis. She's done well with a transplant. It took us a long time to figure out what she had. I would argue that we could tell you what she had within hours now and have a much better approach to her short-term and long-term care. These questions are the ones that are driving our lab. These are the questions that I hope drive much of clinical scientists' thinking. For if we could answer these questions, we would have a tremendously better handle on all sorts of disease. So I want to thank a few people. I want to thank the patients who have helped us. Please understand what our patients are doing. They give us blood with active disease, and I can tell you I've gotten them to be so kind as to give me samples on the first time I've met them in the emergency room. They give us blood every 3 months, whether they're in remission or are relapsing, whether their disease is active or inactive, and without their support, these studies would come to naught. I want to thank you all, the National Institute of Health, for decades now of research support, and I want to thank 3 individuals. Gladys Hirschman, who was the program officer who I dealt with at the beginning of my career, Mike Flessner, who is about to retire, and Marva Moxie-Mims, who I think is here. I know that all of you think from the intramural perspective, there are a number of types of individuals that are important, but for we in the extramural world, where the rubber meets the road is, in fact, with the program officer. So please, next time you run into a program officer who picks you up when you're down, who tells you what on earth is happening on the study section that you can't decipher, give that program officer an enormous hug. I wanted to thank all the other people who helped me on the lab. That was a slide I missed, but you heard about Catherine Huffman Falk, my cohort in crime and Francis's. In reality, in our household, there's no question who's the astute clinician. It's Kathy. And I am entirely indebted to my family support, which you see here. And without further ado, thank you so much for your attention.
Thank you, Ron, for an incredibly elegant presentation. The designation of astute clinician has never set more clearly and well than it has this afternoon. We have time for some questions. There are microphones in the aisles. Please use them so the people listening by video can hear the question. Start here.
So I have 2 questions. The first one is, do patients ever do better on antibiotics?
Oh, of course they do. And I can tell you what kind of antibiotic. And it's that question I raised in the upper respiratory tract. For in fact, I am never sure whether the disease in the upper respiratory tract is an infection or vasculitis, or whether the infection is causing or stimulating the vasculitic event. And there we in fact use a lot of antibiotic topically applied to the nose. My favorite formula is a quart of water with a teaspoon of baking soda, a teaspoon of salt, with a tube of mupirocin, shaken, not stirred.
So do you know if their gut microbiome is different in any way?
Of course, we and others are looking at that exact question. And I'm sure you and others who are doing this realize the incredible complexity of doing microbiome studies in patients who are on and off antibiotics and on and off immunomodulatory drugs, not to mention the other weird things that humans do every day of their life.
Over here.
Yes, would this condition be in any way, manner, or form conducive to treatment with adoptive cell therapy with Tregs that were working properly, as in contrast to the ones in the patient? You said something about the FOXP3 wasn't quite right.
Or should we just apply CAR T cell technology to CAR T? I mean, there are a number of ways of altering T cells that would be fun that we've learned from our oncology colleagues. Yes, of course, I'd love to do cell-based therapy, and at UNC there's a wonderful immune, clinically available approach. The problem there is that we're doing so well in getting people into remission, and in fact are able to take so many patients off all drugs, the question becomes risk-benefit of using a cell-based therapy. But I would love to be able to alter Tregs. I would even more love to do mucosal, to induce mucosal tolerance.
Thank you.
Is there a predominance of female patients in your area of investigation? A lot of antibodies, autoimmune disease are more predominant in female patients.
Not this one. Males and females are equally affected. This is a Caucasian disease more than an African American disease. And it is a disease in some communities in Asia, but not others. But with respect to male and female, with respect to gender, there's absolutely equivalent effect.
You also mentioned there are hemorrhage only for the small vessel. So what is it for the small vessel and why not the large one?
And why the focality in the small vessel? And why does polyarteritis nodosa, for example, associated with hepatitis B, why does that not extend into the small vessel? I would like you to help me answer that question since I have no earthly idea.
Thank you.
Very elegant, very, as Dr. Collins mentioned, very elegant studies. Out of many questions, my question is, you mentioned some differences between the enzyme activation, the two types of enzyme, and the appearance of the disease activation in the upper digestive, is it digestive?
Respiratory.
Okay, respiratory. Right. Is there any differences between composition and activation of different innate immune cells? Because you also mentioned eosinophil, and usually eosinophils are activated upon allergic reaction of mast cell and exhaustion of mast cell.
So there's been some work on exactly that latter question. But the first question you asked me was, why is there a predilection for the upper respiratory tract in patients who are proteinase 3 ANCA when compared to myeloperoxidase ANCA? Right. And I can't answer that question, but I will tell you in data that are going to come out, we can actually create that granulomatosis response in mice treated with myeloperoxidase antibodies by giving LPS in the upper respiratory tract. And in fact, we can make it happen in their nose if we spray LPS in their nose. Not fun to spray LPS in a mouse nose, I will tell you. But in fact, there may be multiple factors, some of which you have alluded to.
Thank you.
Thank you for such a beautiful talk. Do you have any idea why granulomas?
John, thanks. Not truly, but we have just developed an alteration of our mouse model. Exactly the same approach, and have added inhaled LPS, and we get a beautiful granulomatosis response. If we put LPS with anti-MPO in the skin, we get a granulomatosis, not a real granuloma. There's a difference in the skin. We're just at the beginning of those observations. It's got to be the alterations that are now part of innate immunity that are driving that response. But no, I don't know the answer to your question yet.
Thank you.
Do you believe in the hygiene hypothesis, and why or why not?
I think all children should roll in the dirt at all moments. I think dirt is really a good thing to play in as a kid. And I would suggest that with no more data than you have, that we are being so aseptically cautious that growing up in the country with lots of molecules to have to be exposed to is a wonderful thing.
Ron, let me ask, the epigenetic hypothesis that you put forward in terms of what it is that's turning on these autoantigens and neutrophils is intriguing, and yet I would be willing to bet that it's not just that CPG that's getting altered in terms of what happens between a cell that's not risky and one that becomes risky. So have you been able to look more systemically, sort of genome-wide at methylation using a chip of some sort? Or on top of that, is it also possible to look at chromatin structure and see what's really happening more globally to histones that might reflect a wholesale reprogramming of gene expression?
Yeah. So I'm going to do this in two parts. Part one, yes, we have looked at the methylome. And in fact, this is a relatively specific response. It is not broad-based reprogramming, at least in circulating cells. So it is much more specific, not super specific, but more specific than you described. But Francis, why do you think there is actually a alteration in both histone methylation and DNA methylation? What's that telling you? Is there an alteration in, it's not a microRNA, that we know. We don't think it is. Is it a long non-coding RNA? Is it a circular RNA? What is it? Oh, I'm happy to share the podium with you.
Oh, you're so clever. You haven't changed a bit. So, well, one possibility, of course, is that there's some master transcription factor, which is perhaps undergoing some subtle post-translational modification, a phosphorylation or some such other thing that causes it no longer to have the same binding properties. Maybe it's RUNX3, or maybe it's some other factor, which affects the whole program there of gene expression, takes apart a collaboration between various proteins that normally would give a positive or a negative effect on gene expression. And then the methylation follows after it. I tend to think of methylation more as an effect than as a cause. And if you looked at chromatin, I suspect you would see other correlative findings as well. But I'm intrigued that it's as specific as what you say, because oftentimes when one sees those things, it turns out to be much more of a larger program that things have changed, especially if there's a transcription factor that's generally involved in multiple different loci. But maybe you've got a special case, which would be wonderful to unravel. Well, now that I've disrupted the questions, let me thank our speaker and ask you to do so as well. And let's join him in the library for some refreshments.